My name is Tiffany Oliver, and I am a Black scientist. My research involves studying the gene PRDM9 and determining if mutations in this gene is associated with the woman's risk of having a child with Down syndrome. And so answering this question is important because it may help us create a genetic test that would refine doctors' assessment of a woman's risk of having a child with Down syndrome. And so I'm not interested in the entire gene, but rather a small portion called the zinc finger binding domain that determines where recombination is going to happen. And so recombination is important because it helps us put pieces of DNA from our parents together so that we can pass it on to our kids. And this is important because when recombination happens in the wrong place, it can lead to having a child with Down syndrome. So in order to study PRDM9, what we do is we collect DNA from women who have a child with Down syndrome. And so they're most likely to have mutations in our gene of interest. And so once we get DNA from these women, we use PCR to amplify the portion of the gene that actually binds DNA, and then we send it for a sequence. And what we do is we compare that sequence to sequence from women who do not have children with Down syndrome to see if it's different.
Okay. Have you found anything yet?
Actually, we have. So far, what we see is that variation in the DNA binding domain of PRDM9 isn't associated with the placement of where recombination happens, but it is associated with the absence of recombination on chromosome 21. That's the evidence that we find supports that.